Hello students, welcome to SK Biology. Our today's topic is Human Genome Project, Unlocking the Code of Life. Human Genome Project was an international scientific project aimed at determining the complete DNA sequence of human genome and identifying all the genes. Human Genome Project was launched in 1990 and successfully completed in 2003. This project was mainly coordinated by National Institute of Health, Department of Energy, which are government bodies, and one private biotech center called as Celera Genomics, along with contribution of more than 20 institutes from France, Germany, Japan, China, UK, and US. Human Genome Project come up with important objectives as follows: to determine sequence of 3 billion base pairs, to identify all human genes, to store sequencing data in databases, to develop improved sequencing technique, and to address ethical, legal, and social issues. To attain these objectives, scientists worked hard. Now, let us understand method followed by scientists in Human Genome Project. Firstly, they collected blood samples from anonymous volunteer donor. Donors were selected with strict ethical guidelines. From blood, scientists isolated white blood cells as a source of DNA. From these cells, DNA extraction was done by chemical extraction method. After quality check, this DNA is used for DNA sequencing, where firstly isolated DNA is fragmented into small pieces and these fragments are inserted into vectors, BAC or YAC, as shown in figure, vector containing human DNA insert. By inserting DNA fragments into vectors, scientists created library of human genome and stored it for research. From the library, all DNA inserts are sequenced and annotated further by doing bioinformatics study, which led to complete genome sequencing. Fragments used are in the range of 100 to 100, and vectors used in HGP are mainly BAC, that is bacterial artificial chromosome, and YAC, that is yeast artificial chromosome. By following this method, scientists attained milestones in DNA sequencing. Let us discuss them. After starting HGP, first milestone was in 1999, in which chromosome completely sequenced. Then in early 2000, chromosome 21 was sequenced fully. In late 2000, we got full sequence of chromosome X. In the same year later, chromosome Y was sequenced. In 2001, entire draft of human genome was ready, which scientists declared in 2003. In 2006, chromosome 1 was completely sequenced and annotated. After completion of genome sequencing and annotation, the final outcomes of HGP are as follows. First outcome is human genome contains 3 billion base pairs arranged in a precise sequence. Second outcome is human contain total number of gene which is in the range of 25 to 30,000. Chromosome size in human varies from 55 to 250 megabases. Chromosome 1 is largest chromosome and contains maximum number of genes which is 2968. Chromosome Y contains lowest number of genes, that is 231. Human genome is divided into two parts. Non-coding region is 98.5% and coding region is 1.5%. Genome contains large portion of repetitive non-coding DNA sequences called as satellite DNA, which is around 50%. It is mainly found in centromere and telomere. Now, let us discuss some important points from Human Genome Project, on which questions were asked in previous NEET exams. First is expressed sequence tag. These are short sequences of 200 to 800 base pair. Represent Gene expressed as RNA. ESTs help in identifying protein coding regions and used for discovery and mapping of genes. As shown here in figure, genome contains exons and introns. Exons come together to form cDNA, that is complementary DNA. Small sequence on cDNA is expressed sequence tag. Scientists created express sequence tag databases to identify active genes before completion of genome sequencing. Second important point is satellite DNA. Satellite DNA consists of highly repetitive non-coding sequences arranged in genome in heterochromatin region centromere. The key features of satellite DNA are structurally satellite DNA comprises long array of head to tail repeats. They are located in heterochromatin region. Most important feature they have is polymorphism, that is high variation in number of repeats between individual. This feature is useful in research and forensic science for identification. Satellite DNA is of three types: mini satellite, microsatellite and short tandem repeats. Human Genome Project led to the development of bioinformatics. Let us understand role of bioinformatics in Human Genome Project. Bioinformatics is the application of computational tools and techniques to manage and analyze biological information. Bioinformatics played critical role in Human Genome Project for sequence assembly, storage, gene prediction, comparative genomics, protein structure prediction, and managing massive genomic datasets. Without bioinformatics, assembly of billions of base pairs would not have been possible.